CBMNet is an interdisciplinary network in industrial biotechnology and bioenergy. We foster collaborations between academia, industry, policymakers and NGOs in order to find new approaches to tackle research challenges, translate research and deliver key benefits in industrial biotechnology and bioenergy.
Our network is specifically involved in problems associated with crossing biological membranes that might arise in biotechnological processes. Maybe getting substrates into your cell factory or getting products out of your cell factory could be a major problem. And by engineering the membrane function of that cell factory, you may be able to improve the productivity of your systems.
Having a network on the scale of CBMNet is truly novel. With over 700 partners, both academic and industrial, it allows us to move our research towards industrially relevant problems and influence real-world practices.
We have a range of different funding streams available through the network, so we have proof of concept funding and we have business interaction vouchers available. We also have grants for students to attend conferences and also grants for academics to apply for vacation studentships.
At CBMNet, we are committed to helping early career researchers with their career prospects. We have funding available that PhD students and postdocs can apply for to attend relevant training courses and conferences.
CBMNet, being a cross-cutting network, has been a really good example of a fundamental bioscience working with industry to get some really good projects off the ground, which we're now really impressed by the outputs we're seeing through responsive mode, the IB Catalyst and other funding mechanisms.
We are a UK-based company which are developing and commercializing natural N-butanol. When I first met Alan, we figured out that there are quite some synergies between his research and our development program.
I first encountered CBMNet at their first event in September 2014, which was where I first met Preben Krabben, and we worked out we had some research synergies we could carry on and apply for business interaction voucher.
With the help of a CBMNet business interaction voucher, we started working with Goddard looking at artificial plasma membranes and reconstituted plasma membranes, and that has learned us quite a bit about how N-butanol is interacting with the plasma membranes.
The ongoing funding by CBMNet will allow me to continue my collaboration with Green Biologics. From this point onwards, we're looking at applying for large external grants such as KTPs and IB Catalyst to continue the work we're doing.
My role at Fujifilm involves the creation and development of E. coli manufacturing strains. At Fujifilm, we don't often have time to investigate our processes to determine the science that's going on. We developed a project with the CBMNet to examine the effect turgor pressure has on cellular secretion and unwanted leakage.
My group focuses on understanding bacterial physiology parameters, which are like energetics, but it also includes turgor pressure, which is the one that is particularly relevant for this CBMNet project. So we started with a vacation scholarship, and this enabled us to look into and discuss with Fujifilm how can our research, which was fundamental when we started, purely fundamental, be easily and effectively translated into something that is relevant for Fujifilm and overall for industrial biotechnology. We had these two steps. One was the vacation scholarship, and then this enabled us to establish how can we really effectively translate what we're doing and how can we look at this overall whole cell response to changes in turgor pressure into something that is relevant and can improve the processes in industrial biotechnology in terms of getting the periplasmic proteins out of the cell. So through this vacation scholarship, we were able to apply and get Fujifilm on board for a proof of concept award, which then allowed us to really hammer in the kind of questions that are particularly relevant to Fujifilm.
The initial proof of concept project has led to a longer-term collaboration with Dr. Pilazota's group. At Fujifilm, we're hoping to work with many more CBMNet members in the future.
The BIV's been important for us because we had a meeting with Martin Waterfall down at University of Edinburgh, who's got a great flow cytometry service, which is basically a method to screen microbes or cells at a high throughput rate. So in terms of screening capability, which is really important to us, it's got a lot of the attributes that are favourable in terms of throughput rate. The problem for us is at the moment we don't have data or feasibility to demonstrate its application for our strain development we want to do at Ingenza. So the BIVs allowed us to go and work with Martin to evaluate the prospect of implementing it for our microbial strain development for our customers. So it's been really good. And the other reason we want to work with Martin is we don't have the either the experience or the money to go and buy the hardware and then train ourselves to use the equipment. So working with an academic like Martin is the perfect solution for us.
I met Doug Cossar at one of the CBMnet gigs in Sheffield, and I'd not met him before, and he was telling me about a project with an obscure yeast of which I hadn't heard called Pseudozyma aphidis, which makes biosurfactants in quite high levels that are maltoerythrolipids. They had an interest in producing this stuff in enough quantities for industry to be able to test to see if they were any good, but the yields weren't good enough, and so the question was how could you improve the flux to the maltoerythrolipids, and the feeling was that it was likely that the greatest flux control was taking place at the level of the transporters, namely the fatty acid importer. Indeed, we found them, we characterized them bioinformatically, and we put it all together with a nice genome browser so it was easy to analyze the data. It wouldn't have happened without the CBMnet because I wouldn't have met Doug, and it was an excellent relationship that we've continued and put some other bids in together to BBSRC and elsewhere.
T-Gene is a biotech company which is based at Wilton Centre in northeast of England. It's a spin-out company from Teesside University. So I've been working with CBMnet for the past 6 months. I have noticed it's one of the outstanding network among all the NIB network. We have received a proof of concept funding to support the nanomaterial synthesis platform. So currently we are working with the University of York and Johnson Matthey to develop the technology. So this technology will be to produce a platinum group of nanomaterials for commercial applications.
In our case, we've been interested in efflux proteins related to antibiotic resistance, which is a very hot topic in the UK at the moment. But equally, these proteins are important to industry because they use feedstocks like glucose, and the organism converts them to compounds of commercial importance, in our case the synthesis of plastics. So the student also gets interested in projects which are applied, and this is one thing that CBMnet is all about, promoting the interaction between industry and academia.
Funding through CBMnet's been really important for my group. We've built on our BIV award, which worked really well, and we've built up a network of academics and industrialists and put together an application for the BBSRC Industrial Biotechnology Catalyst program. This was successful, and now we have a 5-year program to work with Green Biologics, Lucite, and a number of other companies to try and engineer bacteria to become more tolerant to toxic chemicals that they're manufacturing.
CBMnet have funded many projects in industrial biotechnology and bioenergy, and we're feeling really excited about taking forward the outcomes that have risen from these projects to date. We're looking forward over the next few years to forging more relationships between academia and industry.